I would like to introduce the first presenter, the first talk of today, which is held by Vincent Eijsink. Vincent is a full professor at the Norwegian University of Life Sciences, and his research group focuses on the enzymology of biomass conversion, thereby also covering a broad spectrum of fundamental and applied aspects. And while his current work is more focused towards cellulose and on-cellulose, as you could maybe see on his recent publication records, chitin enzymology has always been and still remains a central research topic for him. The group is well known. Also, that's how I got introduced to your work, Vincent, was by the work on enzyme processivity on chitinases. And he was also the first to describe the boosting effects of certain protein on the efficiency of polysaccharide degradation, which led ultimately to the discovery of Lytic Polysaccharide MonoOxygenases. And Simon, Vincent, I'm really looking forward to hear about the topic and the discovery and all the research along that way. So the floor is yours.
Thank you, Gregor. I'll try to share my screen. Here we go. Perfect. Yeah, so thank you, Gregor, and good morning, everybody, or good afternoon, or good evening, wherever you are. So I will talk a little bit about these Lytic Polysaccharide MonoOxygenases, and my title was How Chitin Helped Change Our Views on Enzymatic Biomass Conversion, because these LPMOs, the discovery of these LPMOs derives from research on chitin enzymology. So let me just see, why is it not? There we go. So we have been interested for a long time in the enzymatic degradation or conversion of recalcitrant polysaccharides. And of course, you all know chitin, shown to the left here. But there's another linear crystalline insoluble polymer out there, which of course is cellulose in the plant world, and which is receiving a lot of attention. Breaking down or doing anything with these polysaccharides is complicated, as shown here for cellulose. There are many hydrogen bonds here that create this very strong crystalline material that has all kinds of interesting properties. But if you want to break it down, for example, this is not so easy. And enzymes that tackle this challenge, and this is shown here for cellulase, later you will see a picture of a chitinase, they face an enormous challenge because, oh, I forgot my pointer, apologies for that. Let me just put in a laser pointer here. So cellulases, like here you have a cellulose fibril with a lot of polysaccharide chains, and hydrolytic enzymes, they have to get hold of a single chain, engulf this chain in its catalytic center, and here, somewhere here, cleavage is then happening. And of course, the decrystallization of this single cellulose chain from the cellulose crystal, that's an energetically challenging task. So enzymes, they do this, and they use, for example, processivity to make this efficient. I'm not going to talk about that today, but we have, as was mentioned, done quite some work on that in the early years, or some 15 years ago. Well, started some 15 years ago. But you can see here that this is an enormous challenge. And in the cellulose field, all the way since the 1950s, and this is a paper by an author called Rees, and you might know this name because the Trichoderma reesi is named after him. And this is about cellulose degradation, but already then people speculated that there must be something more in these enzymatic machineries that break down these polysaccharides, perhaps something like a decrystallizing factor or something like that. And then, as was already mentioned, we came up with these proteins, which they are called LPMOs, Lytic Polysaccharide MonoOxygenases, that perhaps are this factor that was anticipated by Rees in the 1950s. These LPMOs have gotten a lot of attention since they were discovered. They are used all over the world in cellulose processing. Here is a picture from a biorefinery demo plant close to us, a company called Borregaard. And this is a recent paper from 2020, which shows the demonstration scale use of these LPMOs. And we do some stuff here that I'm not going to discuss in detail, but it illustrates that these enzymes, they do very interesting chemistry. So they are very interesting from a scientific point of view, but they are also very interesting from an applied point of view. And if you buy a commercial cellulase cocktail today, there will be LPMOs in that cocktail. Now, let's go a little bit to the timeline. It's actually really, really interesting. Again, we are at cellulose here. This is a paper from 1974 that we did not discover before after 2010. where a group of Swedish researchers, they took a secretome from a fungus, so just, you know, an enzyme cocktail produced from a fungus, and they registered that if they broke down cellulose with that enzyme cocktail, the efficiency of that process was faster in the presence of oxygen. And they conclude from that there must be redox enzymes involved. Now, at the time, the only redox enzyme that it became known approximately at that time was cellobio's dehydrogenase. Today, what they were seeing was probably an effect of these LPMOs, because they need oxygen, as we will discuss in a minute. So this was in 1974. Then in the 1990s, there was a lot of work done by Hildegard Schrempf and her team, and also by Takeshi Watanabe and his team, on these chitin binding proteins. They were classified in the KC database, the carbohydrate active enzyme database, as carbohydrate binding modules family 33, CBM 33. But basically all that was found in the 1990s is that these proteins were everywhere and that they bound chitin. And I'm showing a picture from one of the very nice papers by Takeshi Watanabe and his team. He did a lot of great work on chitin-lytic enzymes until today, but in the 90s he published a few really key papers that are cited a lot. And this is a picture of the secretome of Serratia marcescens. And you see that next to the chitinases and the beta-glucosidase, which is needed to produce monosugars, this produces another protein. And the only property of this protein that Takeshi was able to demonstrate is that, well, two properties. It's co-expressed with chitinases and it binds to chitin. Then I was fortunate to have a PhD student in my group, Gustav Costa, who came and wanted to start working on the CBP21. And in 2005, he published two papers that revealed the structure of this CBP21 and also showed that CBP21 actually acted in concert with chitinases. These are some pictures. Gustav, by the way, won the Brackenau Prize for his thesis. And these two papers were part of that thesis. It's a while ago now, but it's still a good memory. If I remember correctly, it was in Montpellier. I'm not completely sure, but anyway, this is the structure, and I cannot go into detail, but this structure actually revealed some unexpected features that I'm not going to discuss. It kind of suggested that there was something special about this protein. And then right after, Gustav was able to show that if you break down chitin with this is, by the way, a very crappy chitinase, I have to admit. So we took a bad chitinase to make the effect look maximum. And if you then break down with the chitinase alone, it goes very slowly in this case. If you add this CBP21, you get synergy, you get massive synergy. This was in 2005. Then in 2007, there was a paper about another class of proteins or enzymes called glycoside hydrolysis, family 61. They were thought to be hydrolysis because people had seen very weak endoglucanase activity. And in 2007, it was shown that these proteins actually boost well-known cellulosases. So basically, it's a bit the same as what we saw in 2005 for chitin. And even more interesting, in 2008, there was a crystal structure published of GH61, the first one, which showed that this protein and that protein are structurally very similar. And then in 2010, we showed Gustav and a big team of colleagues here showed that these CBP21 and by analogy, of course, also these other proteins are actually enzymes. They are redox enzymes. And the key experiment was the addition of a reductant, so an electron donor to the protein. So here you see chitin treated with CBP21 alone, and with CBP21 and an added reductant. And you see the chitin gets all fluffy. You get considerable degradation of the chitin by only this protein if you feed it with electrons delivered by the reductant. And we also got enormous synergies then with chitinases. 2011 then, as expected, there were multiple studies being published showing that this activity was also found for cellulose, so oxidative degradation of cellulose. And Quinlan et al., Paul Walton's group, they showed something that we had been overlooking in 2010, that the cofactor in these LPMOs is a copper, a single copper ion. And it's important, it's a single copper ion. And as you hopefully know, this means that this protein can store one electron. You can reduce the copper from copper 2 to copper 1. And that's basically what you can do. And this raises issues, as I will discuss in a second. Finally, one more, and I will get back to this in 2016. There was another paper from our group, which I would unmodestly refer to as a landmark paper, where we show that these LPMOs may actually not be monooxygenases, but peroxygenases, and we will get back to that. So what do we know? We have the hydrolysis here in yellow. This is an artist's impression of a cellulose fibril. The hydrolysis that need to engulf a single chain taken out of its crystalline context. And then we have the LPMOs that act on the interface. So they break glycosidic bonds in polysaccharides that are still embedded in their crystalline structure. You also see that here. To do so, they have a single copper there. This is the active site with 2 conserved histidines, and the redox chemistry is happening at this copper. And of course, hydrolytic enzymes, they can, since they can bend the chain, as we'll see in a minute, they don't need as powerful energy chemistry as the LPMOs. So it's not so strange that oxidative chemistry needs to be used to actually do this, to break a glycosidic bond while the substrate is in its crystalline context. Now, what exactly do they do? So this is shown here for cellulose. So you have the beta one for glycosidic bonds, same type of glycosidic bond as in chitin. And the LPMO either acts on this C or on that C. In both cases, you get the hydroxylation, which leads to spontaneous chain cleavage. And if it's a C1 oxidizing enzyme, you'll get a lactone, which will spontaneously convert to an aldonic acid. If it's a C4 oxidizing enzyme, and we see both in nature, then you'll get a ketoaldose, which spontaneously converted to a gamdiol. And this leads to very nice products on, if you have the right chromatography system, you can actually detect these products, both C1 oxidized, because there are also LPMOs that produce mixtures of C1 and C4 oxidized products. And this you see this here. You get a product profile with native products, C1 oxidized products, and C4 oxidized products. Now, when LPMOs acting on the chitin, they only do C1 oxidation. So chitin active LPMOs, they produce aldonic acids. And yeah, I already alluded to this. I want to stress once more that these enzymes really do something very different compared to, for example, a chitinase. From here, you see from a review we wrote in the memory of Kjell Worm early last year. And this is a picture from that review. Here you see chitinase A, which is acting on chitin. And again, you see this single chain that has been taken off its crystalline context, and somewhere here in the catalytic center cleavage is happening. And so the binding energy that you have here between the enzyme and the chain is used to distort the substrate. You can see this here. This is from a crystal structure done by Forgias and others in around the year 2000. So you have distortion here, and this distortion makes it easier to hydrolytically cleave the bond. This distortion is essential, otherwise the reaction wouldn't work, it wouldn't run. If you are acting on a substrate that is in a crystalline context, you can of course not do this. You cannot do this distortion. So these LPMOs do things very, very differently and have to use a much more powerful chemistry, oxidative chemistry, which is a problem. You have to remember, if you do things like this with reactive coppers and things like that, there's a lot that can go wrong. So as in nature, you only use this if you actually need it. And for breaking down crystalline chitin, it's needed. So when we think about LPMOs and chitin, we have, there is in the CASI database, there are currently 6 families. So these are sequence-based families. These proteins are actually all very similar. They look, the catalytic centers, they look very, very similar. But there's a lot of sequence variation, and this gives 6 families. Many of the characterized ones have been, they are involved in cellulose degradation and are only active on cellulose and other plant polysaccharides. And chitin active LPMOs occur in 3 families. The AA10 family, this is the former CBM33 family. It's all the bacterial LPMOs are there, some viral LPMOs. Fungal chitin active LPMOs occur in family AA11. And there are also some chitin active LPMOs in a family with very few characterized members in this called AA15 from algae and insects. Now, what do LPMOs do? So they are going to put an oxygen here. They're going to hydroxylate here, the C1 or C4. And what we published in 2010 is that they need oxygen, they need a reductant, so they need electron delivery, and then somehow there is a reactive oxygen species, so a more oxidative species than O2 is actually thought to be a so-called oxyl, so a very, very powerful oxygen species being generated at the copper site that is able to oxidize this carbon, this non-activated carbon. Now, if in this mechanism, you see for each catalytic cycle, you need, of course, oxygen, and you need electron delivery. So you're going to use stoichiometric amounts of reductant. Every time for each cycle, you need electron delivery. You're oxidizing a reductant. You're taking electrons from a reductant. And early work after 2010 focused very much on this. So where do the LPMOs acting on the surface here, cellulose or chitin, get their electrons from? And it has been shown, which is quite remarkable, that these LPMOs can actually pick their electrons from many, many different sources. From enzymes like cell biomass dehydrogenase, which can convert electrons via the cytochrome domain. It can pick up electrons by chemical reductants, for example, plant and fungal phenols that are, of course, often present when you break down plant biomass. And these compounds can be redox recycled by other redox enzymes. So there's a complex collection of compounds and enzymes that all somehow can affect the redox state of the LPMO, the electron delivery to the LPMO. Now, there is a problem with this, because if you think about what really is happening, then there is a problem which we sometimes refer to as the second electron conundrum. So this is just a modeling study by Bastian Bizarro and Osmond Röhr and colleagues from our university, showing how a chitin-active lytic polysaccharide MonoOxygenases, actually CBP21, binds the chitin. And you see here the LPMO bound on the chitin with water around it. There is a copper site here. Now this copper can store only one electron. The reaction requires 2 electrons. So the question then is, how does the second electron ever get to this copper? Because the copper is completely shielded by the substrate. So while the LPMO is bound to the substrate, somehow an electron needs to be delivered. And it's not easy to envisage how that would take place. And this is why we proposed and also demonstrated in 2016 that it may actually not be like that. It may actually be that these LPMOs do not use oxygen, but that they use hydrogen peroxide. And I'll show you a few experiments that we did to show that. And if this would be the reaction, then once you have a reduced LPMO, the reaction would run without additional supply of electrons along the way, and you would also then not be consuming reductant. Now, what kind of experiments did we do to show this? This is mainly done by Bastian, or almost exclusively done by Bastian Bizarro, a fantastic postdoc we had in the lab. So this is mass spectrometry showing one of the oxidized products. And this is a reaction done in anaerobic conditions. So you would have approximately 250 micromolar of 16 oxygen. And we then added heavy 18 oxygen hydrogen peroxide. And we then saw that the product is actually 18O labeled. And if we start reducing the concentration of hydrogen peroxide even to 25 micromolar, so 10 times less than oxygen, the products are almost exclusively 18O labeled, showing that hydrogen peroxide is actually being used in a reaction and not oxygen. We did a lot of other experiments. Don't try to read this because it will take too much time, but basically this is just to remind me to show you that we then showed that we could inhibit the LPMO reaction if we added another H2O2 consuming enzyme. And we also showed that we could boost the LPMO reaction if we added an H2O2 producing enzyme, like glucose oxidase in this case. So we presented a lot of evidence for this. It's actually still controversial in some groups, not for us, and not for many others. And not at Boku, for example, the Boku team. There is a strong LPMO team at Boku. But they said it was heavily debated. It was not so easy to get these things published, but it seems pretty well established now. And these findings also solved another problem, because you have heard about LPMOs probably. You know they are used in industry. If you look at the rates that have been published for these enzymes, so this is a rate, K, apparent rate constants, for these enzymes, when you use them under these so-called MonoOxygenase conditions, these rates are ridiculously slow. This is the rates of other redox enzymes involved in biomass processing. And these are the rates. This is a rate of 10 to the minus 5 per second. So we are talking about far less than 1 per minute. So this is very, very slow. And what we saw is that when you start running these enzymes with hydrogen peroxide, the rates become more normal. These are the red dots here. And we think that what people have been and still are seeing here in these reactions, where oxygen is the only oxidant present, is actually that these reactions are limited by the generation of hydrogen peroxide, which is very slow in most of these systems. So these rates are not catalytic rates of the enzymes, but these are the rates of H2O2 generation in the reactions. And there's a lot of kinetics being done. It's a great study initiated by Morten Surley and Prit Waljame, who led this, and we did then this proper work with H2O2, and then we suddenly got rates here. You see 6.7 per second, which is a decent rate for an enzyme, and also Kcat over Km values that are decent. So in other words, we think that this kinetic data clearly show that the H2O2 driven reaction is valid, and it's also much more realistic in terms of the rates that are being achieved. Osman and Bastien went on to model this, and I just wanted to show this for your entertainment, because there's no way I can try to explain that. Besides, it should be done by someone, not Osman. But what you see here, this is QMMM calculations combined with experimental data showing Chitin, the catalytic center, the copper ion, and a hydrogen peroxide molecule, and it shows how this could go about. The hydrogen peroxide is very much, let me just start again, is very much distorted by the conserved residues of the catalytic center. You get homolytic cleavage. There's a hydroxyl ion here interacting with the copper. You have an hydroxyl radical, which is strongly confined by the interactions with the substrate and the active site residue. You then get the hydroxyl radical then abstracts a proton from the hydroxyl. Here you see, so you have water here, and you have a copper oxyl, which then abstracts the hydrogen from the substrate, forming a hydroxyl again, which then in what is called the so-called rebound mechanism binds to the radical that has been formed here. And there you go, and here you have your hydroxylated substrate. Now, whether it works like this, we of course cannot really know because we cannot observe this, but all experimental data and all modeling data point into this direction. Yeah, Osman and Bastien then also showed that if you have an activated LPMO, so a reduced LPMO, that it is very much faster oxidized, but it reacts very much faster with hydrogen peroxide than with oxygen. So this sheds, and this is important, now we are getting back to, for example, natural, also Chitin degrading ecosystems. So this is the same picture, and looking at this picture again, knowing what we know now, the question of course is, are all these electron delivering systems, are they really delivering electrons, or are they deciding the rate of H2O2 production in these ecosystems? And actually, when you look at the secretomes of this fungi, going back to this 1974 paper, there is enormous amounts of enzymes going on, there's a lot of redox active enzymes. And of course, when we talk about plant biomass, this is very complex. Don't try to understand this figure. If you think it's interesting, then just look it up. But the point is that in these biomass degrading systems, there are tons of redox enzymes. There is H2O2 being produced. There is redox management going on. So reactive oxygen species are being managed. And this is all connected potentially also to the action of these LPMOs, most likely through the levels of H2O2. I could talk about this for hours, I will not, but just remember that if you're looking at natural Chitin degradation, for example, you should be looking for more than chitinases and LPMOs, also for potential redox partners. So there's a lot to say about this. There's a lot of discussions. It has been claimed that the H2O2 reaction is less specific, or that it leads to additional exceptionally fast enzyme inactivation. We would claim that that's totally not true. And the peroxygenase reaction is now very clearly shown is orders of magnitude faster than this apparent MonoOxygenase reaction. So for us, LPMOs are peroxygenases. We would be ready to change the name, but the field there's still some discussion in the field. If you're interested, you should read the literature, because it's a really, also chemically, of course, this is very interesting, because a monocopper enzyme doing peroxygenase chemistry is actually unprecedented. Okay, before to round off a few examples of recent work that we have been doing on Guyton-active LPMOs. This is a study on the Bacillus Guyton-active LPMO, where Sarah and Seshan, a guest researcher, were able to produce all the variants of a 4-domain LPMO, which gives some very interesting results. Just look at the gray and the orange curves. Basically, it shows that if you the orange curve shows the boosting effect of adding the LPMO to a guytonase degradation reaction. And basically what this graph shows is that if you cleave off the CBM5 or more, in all cases, this LPMO becomes very little active. And this is interesting because the original CBP21 is a single domain LPMO. It's actually in graph E. And you see CBP21 gives a nice boost and it's a single domain. But this LPMO, if you chop off its extra domains, it doesn't work anymore. So you can see that these catalytic domains have really evolved in different directions. This one can do it alone. This one can only do it if the CBM5 is there. That's an interesting observation. We don't really not know what it means, but it's interesting. Another study by Solvanit Makasha and other colleagues in the group showing this is a very peculiar enzyme. It is a one protein which contains both an LPMO and a GH18. It's very efficient in breaking down guytin. This is the red curve. And if you give it reductants, so you activate this domain, it becomes really efficient. It goes like this. So you can see that the LPMO and the guytinase really work together and that you need to activate the LPMO. And if you do this after 6 hours, you get a curve like this. So first it goes slowly and then you add the reductant and the LPMO becomes active and the degradation speeds up, and you will then also see oxidized products. These are just the oxidized products which completely correlate with the efficiency of the product. So your LPMO activity is reflected here and there. So pretty interesting. I have to say though that this is a very special case. It's not so that nature is full of these combined multi-domain enzymes with both an LPMO and a hydrolytic enzyme. A recent study by Yuko Nakagawa, I'm not going to go into detail, but she compared 4 guytin-active LPMOs produced by the same organism and showed that they show different degrees of synergies with different guytinases. Again, we don't know what that means in detail, but it does show that these seemingly overlapping LPMOs, so seemingly overlapping activities, do actually slightly different things in the guytin degradation process. There's a lot more to discover there. About the fungal LPMOs, so in fungi, the most famous LPMOs are these ones, the so-called AA9s. They work on cellulose and they are used in industry, and there's a lot of research going on. But actually, fungi also have a lot of these AA11s, which are guytin active. It's quite decent gene numbers. They are much less characterized, and functionally, it's much, I mean, this whole guytin degradation, LPMO-driven guytin degradation of fungi has not really been investigated a lot. But we show here, and I cannot go into detail, that we cloned one of these. And this LPMO is behaving just like CBP21. It really boosts the conversion of guytin, and it actually also runs very nicely on hydrogen peroxide. So again, it's a peroxygenase. Finally, are all guytin-active LPMOs involved in guytin conversion? There is very many LPMOs in the bacterial world that have been characterized are active on guytin, and often without us understanding why that bacterium would have a guytin-active LPMO. It's very clear from work on guytin-degraded bacteria, making knockout mutants, for example, that LPMOs do play a role in biomass conversion. This has been shown for guytin and for cellulose, of course, also. No time to go into detail, but you make a knockout of the LPMO and the bug grows much less and things like that. So this is very clear. But on the other hand, for example, here we have a virulence factor from Vibrio cholera, a complicated 4-domain protein, which is an LPMO. This domain is an active LPMO, as shown by Jennifer Lohse and Gustav in 2014. We don't know what this means. We don't know what this protein does, but it's very difficult to envision that this protein is involved in guytin conversion. And this is recent work, a beautiful work led by Gustav. And if you want to know more about this, you should really ask Gustav to come and talk with you about the alternative roles of both guytinases and LPMOs. And this is it's a huge study, and it shows very clearly that the LPMO of this pathogen is crucial for virulence, but it does not show what the LPMO actually does. So it doesn't show a substrate. So this remains enigmatic. This is, of course, an incredibly important paper, an incredibly important finding, but this LPMO is active on guytin, but we cannot couple this activity to the phenotype. But the phenotype, well, the phenotype involves activity of the LPMO, but we do not know what the LPMO does in the during the actual infection. But this is, of course, an area of major interest. Okay, to conclude, they are powerful enzymes. They are peroxygenases as far as we are concerned, and quite unique as such. We believe that there is much more to do with LPMOs. We think that the potential has not yet been exploited to the full, not even in cellulose processing. We think there will be novel LPMO functionalities to be discovered, as you saw on the previous slide. And there are plenty of LPMOs in nature. Remember this, they are all over, and there are many of them. So they must be important, and there are likely other functions to be discovered. And of course, chitin research has been crucial for discovering this, and as such also then made major contributions to understanding enzymatic biomass conversion. These are the groups of Svein and me, and also not shown here, the groups of Morten and Osmund. So many people have been involved. It's always risky to highlight, but I highlight here Gustav, who was the main driver in the early phase, and this is Bastian Bizarro, who has done some amazing work in the past 5-6 years that has led to these more recent discoveries. The people here in italics, they are no longer with us. We have a lot of collaborations. I have to go over this because I'm using too much time, but of course we do not do this alone. We collaborate with a lot of different people, have a lot of different funding, have been lucky to be well funded in the past years. There is a lot of reviews being written in the LPMO field. Some people say more reviews than original work. We contribute to these reviews. We also publish quite a bit of original work. So if you want to know more, there is plenty to read. And if you want to know even more and have a good time, you may visit the next carbohydrate bioengineering meeting, which will be physically in 2022 in Oslo. But next week, actually, we have a digital pre-meeting on May 31st and June 1st. Bruno Mersbacher is one of the speakers. It's going to be very interesting. Daniel Kracher is going to speak about LPMOs. And we were supposed to host the third LPMO symposium last October, and this has been postponed till October 22. And with that, I conclude my presentation. Thank you.
Thank you so much, Vincent, for this very exciting talk about LPMOs. It was really nice to see all this history. Maybe a small announcement. So you mentioned Gustav Kolstad. He will be a speaker in the November webinar session, and I'm pretty sure that he's gonna tell us some of the exciting news that you were just sharing.
That's excellent, so, yes.
Are there any questions from the audience? You can simply raise your hand or just like shout out if you want.
I would have one.
Please, Rupert.
Thanks for a very nice talk. Just a general question, maybe I missed it, but when you're measuring digestion or kinetics, this is of course done in suspension, right? Because chitin is not soluble in anything. So you have a chitin powder that is ground to a certain particle size. Does the particle size or purity and all these things influence a lot the outcome of the kinetic studies or chitin source?
Yes, absolutely. This is a very complicated question because the particle size is important. Actually, it's probably more crystallinity. So again, I think it was Yuko, if I remember correctly, we have one paper about that a couple of years ago where with the particle size reduction, you also get crystallinity reduction. So you get different effects. The LPMO becomes less important, the less crystalline the substrate gets. We have shown that, but there is a totally different aspect to this also, is that these substrates, they may be contaminated with, for example, transition metals, which is not a very nice thing because it really messes up your experiments, because if you have transition metals in your solution, you will of course have effects on the generation and also consumption of hydrogen peroxide, with which can also complicate your things. So yes, you have to be, let me put it like that, you have to be very careful about your substrate. And when it comes to product characterization, we normally only characterize the soluble products. As a rule of thumb, you could say that about 50% of the oxidized sites tends to end up on soluble products, but that's only a rule of thumb. So again, you have to be very careful there. So basically the answer is these things are really complicated. And yeah.
Thank you.
So thank you, Rupert. Any other questions? Maybe I take the chance and ask one by myself. Vincent, you mentioned that chitin-acting LPMOs, or should we call them now LPPOs? I'm not quite sure yet. Are they there? So chitin-active LPMOs are only C1 oxidizers. Do you think that's a rule, or is there just no C4 oxidizing LPMO found on chitin? Or is there any effect maybe from the acetamide that makes this happening, or that makes this the favorable reaction?
Yeah, I'm embarrassed to say that I've never really looked at that with the eyes of a chemist, to the extent that I'm a chemist. It may vary. I mean, there are very many chitin-active LPMOs, and it is possible that C4 oxidation has been overlooked or has not yet been seen. But the fact that everybody is seeing exactly the same thing points towards the direction that C1 probably is the only option here. This may very well relate to the chemistry of and the stereochemical restrictions that the acetamide groups pose on the enzymes, but I've never really properly looked at that. But I think it's quite likely. Yeah. But I actually don't know.
Let's see what time will tell us then.
Yeah.
So any other question? I would have several by myself, but then maybe in the interest of time, thank you, Vincent, so much for your contribution. It was really highly interesting. And with that, I would like to move on to Tadeja Katan. It's a real pleasure. She volunteered to present her master thesis today. Tadeja is currently a master student at the Institute of Chemistry and Technology of Bio-Based Systems at Graz University of Technology under the supervision of Rupert Kagel, who just contributed to this webinar last month. She obtained her bachelor's degree at the University of Maribor before she moved to TU Graz to her master's to Austria. And her research activities cover the modification of surfaces with a focus on chitosan thin films. And there she's concentrating on functionalization of biomolecules, which includes the immobilization of glycopeptides. And today, that's what you're going to talk about today. I'm really keen on the details that Rupert has outlined already last month. And please go ahead.
Thank you very much. Just a moment. Is it working?
Yeah, it's working. Just need to go to presentation mode.
Yep. Moment.
And then you're set.
Okay.
Perfect.
So hello, everybody. My name is Tadeja, and today I'll be presenting a part of my master thesis, which is regarding chitosan thin films and the synthesis of peptidoglycan on their surface, like a surface modification. So the content of today's talk will consist out of aim of my thesis, theoretical background regarding thin films, experimental work, some results, and outlook of the work. So to start off with the aim of the thesis, the aim is to make polymeric thin films, which will be subsequently functionalized by coupling them with certain amino acid and creating a peptidoglycan mimetic, which you can probably already see from the picture above. So to start off with some theory, the in-theory thickness of the thin films ranges from a fraction of nanometers to several micrometers, and there are several methods for thin film deposition. Amongst them is also spin coating, which was the preferred method that we used. The applications of thin films have been found in many fields, but the field that we are most interested in is biomedicine, where they could be maybe used as implant coatings. Moving on to experimental work. To prepare thin films, we firstly had to prepare a solution, which was made out of low molecular weight chitosan, and it was dissolved in acidic aqueous solution. The solution was then deposited onto a substrate, which was either silicon wafer, which you can see on the bottom right corner, from which the 15 to 15 square millimeters substrates were cut out. On the other hand, I also used gold quartz crystal, which you can see in the top right corner. And the QCM crystals are mainly used with the quartz crystal microbalance method for measuring precise masses, which I'll describe a bit later on. So, as I already mentioned, we use spin coating as for the production of uniform thin films. And a spin coater can be used to produce very thin films. And in this method, certain amount of liquid or solution is deposited in the center of this substrate and can be spun from 10 rotations per minute to up to 8,000 rotations per minute. And with this centripetal force, we then create an even covering of our substrate, which can be either silicon wafer or the QCM crystal, as I mentioned before. But with adjusting a lot of parameters, you can certainly play around with what kind of films you get. So we had at the beginning a lot of challenges of producing such uniform films that you see now in the green bracket. For example, one of them was that the film wasn't dispersing, or it wasn't covering all the substrate, or it forms such uneven, or because of this uneven absorption on the substrate, we get such spots, or even a comet streaks, such as are seen in the third picture. After we prepared this Chitosan film, we then had to neutralize it, because without neutralization, our film would easily dissolve in water. And with this neutralization step, we then achieved that the film was actually stable. And from the pictures here, you can already see the difference in color from an empty substrate to coated substrate and then to neutralized substrate. And the difference here is that the color after neutralization turns slightly lighter, because we presume that some amount of film was actually already lost with this step. So after we established and produced these stable Chitosan thin films on either silicon wafers or gold or QCM crystals, we then moved to characterization methods. And one of them was the atomic force microscopy. We compared the bare uncoated gold crystal to Chitosan film crystal and then to neutralized Chitosan film. And as you can see, there is some kind of some difference when comparing Chitosan native to Chitosan neutralized. You can see that the particles are getting a bit bigger. And this is also observed from the roughness measurements, because the roughness of the neutralized film is slightly higher than from the native film. And as we already seen from the pictures of wafers, that the color got a bit lighter. This was also confirmed with thickness measurements, where the Chitosan native thickness was approximately 44 nanometers. And after neutralization, this thickness dropped for about 25% to 33 nanometers. And how did this neutralization step affect the wettability? So as you know, if the water contact angle with your surface is quite low, this means that the surface is very hydrophilic. And with the case of Chitosan native film, the contact angle was rather low. And after the neutralization step, the contact angle got to about 50 degrees, which gives us a bit, not so much hydrophilic surface afterwards, but still hydrophilic. Chemical composition. So the XPS measurements were done with cooperation with Institute of Josefa Stefana in Ljubljana. And they gave us these excellent results from our native and neutralized types of film. And from the C1s peaks from XPS measurements, we didn't see any drastic changes in the carbon-carbon bonding. However, we did find that in the nitrogen peaks, we could observe that the protonated nitrogen area dropped from approximately 40% in this case down to 4% after neutralization, which concluded and confirmed that the neutralization step of our surface actually worked. The surface elemental composition was also calculated and measured, and the results were rather similar, once again proving that the chitosan was actually on the surface. So now I wanted to present to you the quartz crystal microbalance with dissipation because of its importance in the further research that I've done. So this is the instrument I'm talking about. It's called QCM. It consists out of 4 chambers that you can see here. And in each of these chambers, you can put 1 QCM crystal, which looks like that. And it works upon a piezoelectric effect, which means that if you apply voltage, the crystal starts to oscillate at a certain frequency. And with this frequency change, you can then measure what amount of mass was deposited on your surface. And with the dissipation function, you can also measure the damping of your surface. So for example, if your surface was very soft or very rigid, you would see it with this measurement. So with that in mind, we then measured the mass of the film that was coated on our QCM crystal. And to start it off, we did a baseline measurement of an empty crystal where you can see the frequency is 0. Then after chitosan film was deposited, the frequency shifted to a -360 Hertz, which corresponds to 6.3 micrograms per square centimeter and can be calculated using this Sauer equation. After the neutralization step, we observed that there was some amount of mass that was actually lost, which was already confirmed with the thickness measurements and already by simply looking at the wafer. Afterwards, we then tested the stability in water of our film. So we wanted to compare the starting mass of our film with the end mass of our film after exposing it to water for a certain amount of time. And from the measurement, we concluded that because the film mass at the end was clearly the same as at the beginning, that our film is actually stable in water and didn't dissolve. Afterwards, we did some varying pH value measurements in which we exposed our film to a range of different pHs, starting with pH 7. And some drastic changes started to happen at the pH, at the borderline of pH 5 to 4. You can observe here a sudden shift in frequency to higher values, which would suggest that our film is being protonated and then suddenly dissolved at the pH of 4. And after measuring the dry mass, comparing it to an empty crystal at the beginning, we see that there is pretty much none of the film left on the surface. So moving on to functionalization, or we wanted to try if our film could be digested by lysozyme, which is an enzyme, and by introducing our film to the lysozyme buffer solution, we see here that the frequency shifted to higher frequencies, meaning that some mass was deposited on our surface rather than dissolving or digesting our surface. So in this case, the lysozyme didn't digest our surface, it rather bounded on the surface somehow. But this is quite a recent experiment, and some more characterization methods and analytics had to be done to confirm that. What we actually did is bind proteins or amino acid to the surface of our chitosan film. So this was a concept that was already proven in many literature reviews that we looked up, but it was not so much the solid substrate. the synthesis on solid substrate was not very well represented, so this is something that we wanted to try out. So we coupled the protected amino acid, bok-glycine, with the coupling reagent carbodiamide and measured the change in mass after the coupling reaction inside the QCM. So the change that we observe is that the mass from our film to functionalized film rose for about 2 micrograms per square centimeter. So with that, it is a step in the right direction, that something is actually happening on the surface and something is being bound to it. So one of the things that we tried out with this functionalized surface is that we tried the same experiment with the different pHs and try to see if it would still dissolve here. And as you can see, the same thing happens. So from that, we can conclude maybe that not all nitrogens have been properly functionalized, and still there are still some who have been protonated and could maybe dissolve the film even after functionalization. With the morphology images, we once again see that the particle size gets even higher or even larger with functionalization. So there are some surface changes that we can observe with AFM images, which is also confirmed with the roughness, which gets higher. And from the phase images, you can still see that the film is pretty much intact and that there are no holes or any other damages done to it. It's still quite uniform after functionalization. With attenuated total reflectance, we then measured some ER spectra. We compared the native film with the neutralized, and the purple line represents the functionalized film. And at the 1600, you can see the appearance of a new peak, which could maybe be attributed to a formation of new amide bond in our functionalized film. So to confirm that, we then measured XPS, and from the carbon-carbon bond from the carbon-carbon bonds of neutralized film compared to carbon-carbon bonds of the functionalized film, you can see that the area has risen from an 18% to a 33% in area, which once again points toward the formation or the binding of this bok-glycine protective amino acid on the surface of our film. This was also confirmed with the surface elemental composition, where the ratio stays the same for the actual measurements as well as for the theoretical composition that we calculated. In summary, so all in all, we were able to produce stable chitosan films. With the XPS measurements, we can show that our film can be protonated and with protonated, and with neutralization then deprotonated. We can successfully measure the dry mass of our films inside QCM. And the functionalization of the chitosan films is also possible. So for the outlook of this work is that the knowledge that we now obtained from the chitosan films, we want to transfer to synthetic films maybe, and proceed the same kind of procedure to make these films of PVDF, for example, and then subsequently functionalize them with a range or with a different amino acid. And if we succeeded that, we can maybe then use such coatings in the biomedical application. So thank you very much. This was all of my presentation. And if you have any questions, I will try my best to answer them.
Thank you so much, Tadeja, for this really interesting talk. Really well done. Are there any questions from the audience? Vincent? Or sorry, that was just a clap, right? I am still...
Yeah, it was just a clap, yes, yes.
I'm still very new in this emoji thing on Zoom. Sorry. Yeah, there is a question. Please go ahead.
So thank you for your kind presentation. And I have a question about how did you prepare the chitosan from the chitin?
How did I obtain chitosan from chitin?
Yes,
Yeah.
is the commercial or you used any MS?
No, it was the commercial bought from Sigma Aldrich, low molecular weight, commercial.
Okay, thank you.
So thank you. Is there any other question? Maybe I can take my chance again. Tadeja, maybe I totally missed it, but how did you neutralize the film, the chitosan film?
We used an alkaline solution. The film was basically, we used sodium hydroxide solution. So by immersing it in this solution for a certain amount of time, and then washing and drying, measuring again, we neutralized the surface. Yeah.
Okay. At which pH did you perform the this glycine coupling, like the amide coupling, the carbodiamide coupling? I think you mentioned. At which pH was that performed, and which implication does that have on the whole immobilization efficiency and so on?
We tried it with the lower pH, and it did not work as well. So we then went on with the pH of about 9, and this is where the most successful results were obtained from. So
okay, really interesting. Are there any further questions? Bruno? Or was that a hand raise for a question, Jan?
Yes, it was. Sorry that Vodafone decided today that I don't want to join these seminars. I keep losing the contact always.
Oh, I see.
I didn't hear all of the talk. But I have a question also to this neutralizing step, because I think you said that you lose about 20% or so of your material when you neutralize.
Yeah.
And of course, when you start, the chitosan you use will be a mixture of different molecular weights and also different degrees of acetylation. Whether you have a loss that is dependent perhaps on dP or dA? I mean, like small molecules lost than large ones, or the other way around?
It could be. We didn't do any experimental comparison of that, but it could be maybe that due to that lower molecular weight or higher molecular weight are lost. I'm not quite sure.
So we can't tell you.
Yeah, we did some experiments by neutralizing it also in the ethanol, so with ethanol rather than water, and maybe this would yield better results because it doesn't dissolve as well in that. So this is something that we maybe have to look into. Otherwise, yeah, it could be what you suggested.
But the elemental composition did not change, or did it?
No, it did not change.
So that might argue against loss of a specific high or low degree of acetylation.
Okay.
Because the degree of acetylation would influence the composition, the molecular composition, the atomic composition. Okay, thank you.
I think that also highly depends on the incubation time of sodium hydroxide on the films. So I could imagine that would also already give you another hint. Maybe if you like incubate it longer in the sodium hydroxide solution, you could presumably have more de-N-acetylation that you would just have with a fast flush.
Okay.
Great. Yeah, with that, I would, if there are no further questions from the audience, would like to close the webinar for today. Thanks to everyone for listening to it. Thanks again to the presenters. It was really exciting, both topics. Learned a lot again. And we see each other next month. I'm missing the date now. I think it's the 29th of June. Yeah. including a contribution of Thomas Hahn about topping chitin extraction and production from non-marine sources. So there's going to be some exciting news on that side too. Thanks everyone, and have a good day.
Thank you. Bye.
Bye. Thanks.